आ गए हैं अभी वही हो साउंड हो रहा था। हाँ रिकॉर्डिंग ऑन है। हो हाँ ठीक चल रहा है। नहीं कोई दिक्कत नहीं, शोर ले रहा है, थोड़ा सा क्वालिटी कम है। हाँ, ठीक है। To an enlightening session that uses epigenetic and oncology in ways that promise hope for innovative cancer therapy. Thank you. ठीक है अच्छा अच्छा Shall we start? It is my honor that Dr. Santosh invite me to talk about epigenetics on cancer. So I'm working on the cancer, I think from last 10 years. But few of the paper published on the epigenetics and remaining are only on the pure immunology. Okay. So can you slide full page? अच्छा सारे भाई okay. So I think from last 1 week, you people studied about epigenetics, its basics, right? So everything is now clear. So I will not talk much about epigenetic modification, its basics, what are the factors. So we will just remind previous stuff what Professor क्या नाम है Kunal Rai. Yeah. I met him also in MD Anderson because Baylor College of Medicine and MD Anderson is just side by side. So cancer as an epigenetics, let's begin its by history. How many people know him? Have you read any time? No. So like he is the first scientist who discovered basically, but not recognized during that time. People thought that this is impossible. Like in the current scenario, we know that we can talk by mobile without any wire, but 20 or 30 years back, people don't believe that we can talk like this, and we can visually talk as well. So same thing was happened with him as well. But really he invented in 1920, and first time the term is given by the Waddington Quine, the epigenetics. So I hope epigenetic modifications, now you people understand what is the DNA, histone, everything. Other than that, what are the things can also regulate the transcription of gene? Can anyone tell? There are several factors. Okay. Huh.
Transcription factors affect the transcription, then the phosphorylation, methylation at different sites, acetylation on the DNA.
They are all histone modification. Other than that,
we have enhancers, super enhancers,
yes,
promoters. These elements also affect the rate of transcription.
Yeah. So Kunal sir also teach those things. Wow, good. But few of things I will also cover, because lines, signs, you know, and enhancers, super enhancers, suppressor, they all play role in the gene regulation, gene expression. And all this disease is appearing because of sometime over suppression or over expression, uncontrolled expression. Okay. So what basically is the cancer? No, just terminology of the cancer. Uncontrolled cell growth. That is the cancer. There is a no control. They don't know how to stop. So they are continuous growing and making the cells. But in our body, everything has switch on, switch off stuffs, right? When you eat any food, your digestive enzyme start to secrete. They are not secreting all the time. and to start there is a trigger, to stop there is a trigger. Okay, so like in our body several dangerous stuff is also made, like HCl, where stomach, its pH is how much? Can you hold on hand HCl? No, but our stomach holds. So every day our intestine get renewed after every digestion. So that's why if you make the routine for your food habit and that is getting changed, people get acidity or problem in digestion. So our body cycle always understand when we are going to eat the lunch, when we are going to eat the dinner. So during that time you feel the hungry. It is not like that you feel any time. So that particular time usually you feel, right? Because your machinery calculate everything. Like these days we are reading the using the AI, but our body is itself is AI. It's know how, your factors, your understandings, and even your behavior. So it prepare everything accordingly. So even though in 10 individuals, they have all different, their thinking, their stuff. So their body structure is also changed accordingly. Okay, let's begin then. So short summary is like introduction to epigenetics in cancer. Genetically alteration in cancer was normal cells, methylation, histone modification, full epigenomes. Okay, here one terminology came, epigenome. What is epigenome? Huh? On the genome. More elaborate, anyone? Epigenetic and epigenome. These are two different terminology. Yeah.
Or epigenetics are factors which are not in the sequence of the DNA, but they're added onto the DNA, which change the expression of the same.
Someone more about epigenome? It is getting closer. Okay. Okay. See, there is a one major difference is that epigenetic we are talking at one place. Epigenome is we are talking whole chromosome, whole genome, full chromosome is like modified by the epigenetics. So that makes one. Have you read the holes in the cheese alignment? That is the system is happening. So if that all alignment of epigenetic modifications clustered together at a certain time, then it is the initiation of the disease development. It is not only cancer, it can be any neurological disorder, it can be any autoimmune diseases as well. So there are several diseases we don't know the etiology, how they starts, like EAE, some autoimmune diseases like on the skin, and like IBD. I will talk little bit about IBD on the last lecture, in the last lecture. Okay, and some more examples, and what we are doing right now on leukemia, that will also I will talk. So but before that we will understand the basics, how they starts. Okay, and so can we use the epigenetic understanding or epigenetic machinery for the therapeutic aspects? Yes, because epigenetic is not the modification in DNA sequences. It is just modification on the expression of particular genes. We are not mutating, we are not inserting anything, but we are controlling. ठीक है, जैसे किसी को भी पीएम बना दिया या फिर कुछ बना दिया, but he don't have power to do anything. Everything is doing by the secretary, right? Same thing is happen. Secretary will tell that today we have the meeting, today we have to travel there. Okay, so by analyzing the epigenetic markers we can identify the cancer cell markers, tumor prognosis, chemotherapeutic sensitivity, some future ideas in cancer epigenome as well. Okay. So this you people already read? Wow. So like full 10-day epigenetics is going on. So maybe possible there is a overlapping stuff. Okay. So like you can see our lifestyle, our food, diet, aging, environment, all they can make the changes into the epigenetic alterations. Okay. And that is creating the genome instability. That is the main reason is normal cell can convert into the cancerous cell. Many other diseases also linked with the epigenetics. I already told you people, but now my role is here for today's is teaching on the cancer. Okay, because I'm working on the three aspects, even microbiome also. Okay, transcriptional addiction in the cancer. What is the addiction? हाँ ठीक है ओके एडिक्शन जैसे इन नॉर्मल सी एवरी आवर जीन रेगुलेशन एवरीथिंग इज you can find the example in your daily life as well. Addiction is same. Okay, so cancer arises from genetic alterations that invariably lead to dysregulated transcriptional programs. These dysregulated programs can cause cancer cells to become highly dependent on certain regulatory of gene expression. अभी हम लोगों ने इसी बारे में ओके सो एवरीवन अंडरस्टैंड हिंदी एंड इंग्लिश ओके because sometime I switch. Okay, so so transcriptional control if disrupted, can you read the four line? By genetic alteration in cancers, by transcriptional dependent can develop an consequence of the dysregulated programs and how these dependencies provide opportunity for novel therapeutic intervention in cancer. If we understand all these mechanisms, then it's very easy to find out the therapies. So because of that, all this is going on. Okay, plasticity, elastic and plastic. You people understand, right? Okay, good. I have good audience. Epigenetic plasticity and the hallmark of the cancer. So I think you people understand this slide. So let's move next. This figure also you people understand properly. Okay. Environmental factor on gene expression in DNA. The molecules is called the methylation control when gene are turned on. Methylation turned off gene, acetylation turned genes on. Acetylation is happening on DNA or histone or both? Both है ना? Methylation both. If acetylation is increased, what is going to increase? Can it reduce also? Huh? Okay. Okay. So it is a universal in methylation? Yeah. Okay. Good. I think sir teaches very well. Everything. Now my role is very easy. Meiotically and mitotically and meiotically. Have you? This is just simple. Mitosis and meiosis. Mitosis is happening in which type of cells? And meiosis? Okay. So if I say mutation or like epigenetic modification happens in the mitosis, mitotically active cells, can it pass to the next generation meiotically? Okay. Any example of those diseases in both cases? In cancer? Skin cancer? Anything. Okay. And meiotically, ये थोड़ा डिफिकल्ट है ना, जर्म लाइन में चेंजेस होना. अच्छा. ओके. तो CRC, हाँ, hemophilia can be possible. Diabetes also sometimes is goes. Other than cancer, you people can take. And a few cases found in case of leukemia also it's prone. CRC happening in which organ? What is CRC? हाँ, colon, colorectal carcinoma है ना. Okay. So usually that's why my one study is going on the CRC as well. Around 10% patient of the IBD can turn to the CRC, especially in case of ulcerative colitis, because that is only affecting the colon portion. Okay. And one new study is going on, fibrosis in the colon. What is the fibrosis? फाइब्रोसिस और नेक्रोसिस में क्या अंतर है कुछ? हाँ, फाइब्रोसिस में. Okay. Okay, good. At least you people know something. Okay. So fibrosis is also linked with epigenetic modification changes. Okay. And like molecule modification in chromatin is DNA methylation, you people know. Histone modification on H3H4, acetylation, phosphorylation and methylation. So acetylation and phosphorylation is happening only on histone or other proteins also? This comes in the post-translational modification. So in acetylation, acetate group attached, methylation, methyl, and there is a phosphorylation. This is also kind of post-translational modification. Usually that is happening in case of enzymes or signaling cascades. ठीक है ना. So sometime switch on or switch off is dependent on the phosphorylation. So like sir already told me that if he shows the gel picture it's difficult to understand, right? But phosphorylation usually people identify by the protein gel, है ना. Okay. And here you can see ncRNAs, non-coding RNA, microRNA and siRNA comes in that domain. Chromatin remodeling, protein complexes and SNP. So okay, these days microRNA and siRNA is also in quite hot field into the research. Not recent, it is going on from last 10 years to 15 years, because people think that they also play role into the gene expression regulation. Okay. तो ये तो अभी पता ही होगा, पहले ट्रांसक्रिप्शन होता है फिर ट्रांसलेशन होता है. ठीक है. सो मोस्ट ऑफ द मॉडिफिकेशन हैपनिंग एट द पॉइंट ऑफ ट्रांसक्रिप्शन. And after translation, just now we discuss about post-translational modifications. Okay. This figure already sir shows you. Yes. There are not many figures. Only this kind of figure is available. So heterochromatin, euchromatin. So I think you people studied already in high school, in inter. Okay. So for any gene expression, euchromatin structure is needed. In heterochromatin, they cannot. Okay. So, have you people already know that like hypomethylation, hypermethylation? Okay. And you can see here lying in between, क्या है ये? ncRNA, non-coding RNAs. So like in this way, they make the triplet of the, usually DNA is duplex, right? So they can bind and they can stop the gene expression by this way as well. So ncRNA is can be linear as well, can be circular also. It is not necessary they are always in the same structure. So which one is a more stable, linear or circular? कंफ्यूजन है सब में, सर्कुलर ज्यादा होगा ना. ओके ओके. Epigenetic markers in the normal and cancer cells. Marks. So dependent on the cell type, cell and tissue type. So all tissue type like your liver cancer, skin cancer, breast cancer or brain cancer, all they are different type of tissues. So because of that, regulation is also changes accordingly the tissue type. Okay. So DNA methylation, CpG isolate, transcription starting sites, UTR's area, hypo and hypermethylation, this already you people read. Imprinting, X inactivation, hypermethylation, repetitive sequences, transposable element and hypermethylation. Histone modification में acetylation, methylation, phosphorylation, ubiquitination, sumoylation. Ubiquitination, huh? Where it's bind in case of epigenetic. If we, okay, and what is the role of this? Okay, and what is the final outcome of the ubiquitination? Degradation. So sometime like when cell try to divide, during that time usually degrade, because during that time it's need to be the actively replication, duplication of the chromosome, every chromosome, right? ठीक है. So during that time it's need to be full-fledged replication of whole chromosome, every chromosome. Okay, ठीक है. So but they are also playing role in case of gene expression. So like few housekeeping genes in our body, in our cells, they are always active. If they switch off, maybe cell can die. Like for what type of processes needed, always gene should be active. Huh? Yeah, like production of enzyme, you can say metabolic gene. Metabolism is always going on, like glycolysis, TCA cycle, actin filament. ठीक है. ER, mitochondrial. Okay, one thing came in my mind. Does mitochondria has its self genome? Okay. Uh, if we talk about evolution, how evolution make the mitochondria in eukaryote cells? Which type of bacteria? Aerobic or anaerobic? Anaerobic? Aerobic. Okay. Chalo, ठीक. Same thing happens in case of plant cells. There is a Other than mitochondria, there is a one more machinery, chloroplast. What type of bacteria? Photosynthetic bacteria. Okay, chalo. Somolysis is also kind of ubiquitination. So इसका sumo complex बनता है, ठीक है ना? So every word has different name. I don't remember. S U U means like sumo. U means ubiquitination. So ubiquitination like protein. Okay. So until now we was talking about epigenetic modification. Now here you can say that just now we discuss about enhancer and super enhancer and signaling to enhancer, how it happens. So like few example is already I can tell you. Here is given the some enhancer LIF, TGF beta, WNT. They are transcription factor. Okay, they go and bind on the enhancer sites. So enhancer is basically is the DNA portion. Okay, so like by this way a particular gene's expression we can increase several fold, but in some cases like in immune cells we always try some cytokine production is tremendously increased around 100-fold. How it happens? There is another kind of regulation in our body is copy number also. Few genes has several copy number. So if copy number increase 1 to 5 and one gene expression is started from 1 to 10 fold, then total how many fold it's increased? 50 fold, because we have the 5 copies. So in few gene cases we have the our body also regulate by the copy number as well. Okay, so you can also see few people are super active. Why? It may be possible from their office parents they got more copy numbers. So those character usually pass on in the generation as well. Okay, so why they call the enhancer and in which scenario they call the super enhancer? So enhancer, one transcription factor on one enhancer site is regulating. If multiple transcription factor align together and they are controlling the gene expression, then it's become super enhancers as well. And enhancer sites may be possible, not only one, may be possible there is a 3, 4. Sometimes they make the loop also. Maybe sir already teach those things, right? Remember? Okay. So like 3 transcription factor you can see, STAT3, SMAD3, TCF3. When they are all together at the same places, and even LIF, TGF beta, WNT, they pull on the same site, then they are become acting as the super enhancer. And if they are individually, then they just call the enhancer. Okay. इस स्लाइड में और कुछ है क्यों नहीं? Okay. So now we are going to try to understand how a normal cell turning to the cancer cells. So epigenetic changes can make the primed. Primed means like, yeah. And again epigenetic change can lead to the to move cancerous side, but not real. But if there is a DNA alteration and these two things coupled together, like in this case you can say cancer initiation started. So because of that, like single factor usually not governing towards the development of a disease. Multiple factor aligns. So that is also one type of control in our body. It is not just dragging by one factor. Otherwise, everyone will get the disease. So all these three scenarios is turning to the cancer progression and metastasis. So, but you can see usually cancer starts from one cell. I think in second class, I will teach you what is the difference between core cancer cells and there is some specific cancer cells. There is a terminology called the stem cancer cells. Have you people read this terminology? Okay, so in detail mechanism we will go in second class. But for the time being, we are going to understand epigenetic and cancer, how they are related. Okay, I think still many slides remain. Okay, so various epigenetic elements like there are several paper back to back came. Differential DNA repair underlines mutations, hot spots in active promoters in the cancer genomes. So you people have already the basic understanding of the this when mutation is accumulated for a particular oncogene on the promoter side or enhancer side or suppressor side, they can lead to the disruption. Okay, but not only one places, multiple gene if affected, then in that case cell is going to cancer develop. Otherwise they go for apoptosis. CTCF cohesin binding sites are frequently mutated in cancers, disruption of chromatin folding domains. So like here you can see one more words, chromatin folding. क्रोमेटिन पता है क्या होता है? हाँ. So if folding is not proper, when cell is dividing, folding is not proper, in that case also they can be become cancerous. Okay, folding domains by somatic genomic rearrangement in human cancers. So we have very fantastic mechanism into the cells to stop to development of the cancer, like repair mechanism. If cell somehow unable to repair, then cell is undergoing for the apoptosis, self destruction. है ना? Best word is that. And if there is any protein refolding or something is happen, then where they go? For ubiquitination, for degradation. So don't think that your body is making only 1 or 100 proteins. Every minute, every second there is a thousand protein is made, but most of the protein is undergoing for the degradation as well if they are not properly folded. है ना? आप कोई भी चीज बनाते हो हमेशा सेम क्लोन नहीं बनती. कंप्यूटर इज नहीं इज ओनली इन कंप्यूटर इट इज पॉसिबल टू कॉपी एंड पेस्ट. Otherwise, if you are making anything by mechanically or by hand, not possible replicate. So because of that, several checkpoints is there to stop these accumulation of this kind of aberrant processes. Okay. Some example I will give. Super enhancer. There is a kind of DNA break can increase the expression of oncogene. And DNA break near to the enhancer and insertion of the MTF binding sites between two enhancers. And here you can see that normal gene case, they make the loop and enhancer can control the normal gene expression. But in cancer cases, the loop is not properly made, and because of some mutated anchors, so they cannot control. This is a kind of a super enhancer or enhancers for mixed gene regulation. So you enhancer can be present either side of the promoter, positive side, negative side also. And okay, before that, moving more forward, have you people think that bacteria also have the epigenetic modification. This we all talking about always for the eukaryotes. In prokaryote, does it possible? क्रोमोसोम होता है तो क्या होता है? देखो, हिस्टोन मॉडिफिकेशन और फोल्डिंग, एवरीथिंग इज नीड द हिस्टोन, राइट? विदाउट प्रॉपर, अभी हेट्रोक्रोमेटिन पढ़ा हमने. हेट्रोक्रोमेटिन इज वेरी इंपॉर्टेंट टू मेक अ क्रोमोसोम. If there is no chromosome in bacteria, epigenetic modification is possible or not? तो उसमें कैसे कंट्रोल होता है? Anyone? Any idea? Okay. Just think that if there is a no histone, so only histone is not making the epigenetic modification. Other than histone is also, what are there? Some more clues. हाँ. Okay, DNA methylation, acetylation can be possible. Phosphorylation DNA में होता है, तो हिस्टोन में होता है ना. अभी तो पढ़ा है पोस्ट ट्रांसलेशनल मॉडिफिकेशन. ठीक. So those kind of mechanism is present also in bacteria to control a gene expression. Okay. There is a basic one more differences is there. Bacterial chromosome usually have the oric site, origin site. But in our cases, there is a gene is also clustered. We are having the, in particular, in every genes, exons and introns. Those intron can also have some enhancer as well or suppressor. But in bacteria, there is a no exon or intron. Intron. ठीक है? And in bacteria, there is a one more difference, big differences. In human being, if we have 5 kb gene, 5 kb DNA sequences, kilobase, and if I give you one condition, one gene can be have 1 kb kilobase pair. How many gene can be accommodate? 5. In bacteria, if I give same 5 kb total DNA and each gene will having 1 kb, it can be possible less or more. Why? Yeah. Okay. Because bacteria always have the several overlapping genes. It does not have the intron as well. So just think that 10 kb and there are 1 kb gene, but from one first gene, in middle of the first gene also, second gene can be started. So like this, there is a one controlling site in bacteria can control several gene expression together. Because bacteria is very simple organism. It is kind of in evolution first cell, right? So it makes its mechanism very simple. जैसे ट्रेन में चलती है तो एक ही इंजन होता है ना, सेवरल डिब्बे हो सकते हैं, लेकिन जब एरोप्लेन चलता है तो उसमें सब कंबाइंड ही एक ही होता है. So in several processes, same like several cluster of genes they need to process. Like in metabolism, if 5 gene enzymes needed, so it is not like that you are expressing each gene separately. So there one controlling site express all 5 gene together, so that process can be finished. But in human case, all cluster separately. Even not only chromosome, like on the same chromosome, they can be different places, but on the different chromosome also. ठीक है? So if they are on different chromosome, that is another controlling mechanism inherited came. Okay. Usually this kind of data, like you can see the bars, what is they are. Huh? Okay. Chipseek. Yeah. So initially the technique when evolved first time, it was called the chip, then chipseek. Now it is called the ataxic. है ना? तो these things are developing so fast. कभी आप लोगों ने देखा है कि जो डीएनए का सीक्वेंस करते हैं वो पहले कैसे किया करते थे? आजकल तो इल्यूमिना आ गया है ना? एनएसजी आ गया है, एनजीएस, नॉट एनएसजी, एनजीएस, नेक्स्ट जनरेशन सीक्वेंसिंग. At the initial time, I think I also did for something. People was running this much big gel, ATGC. 4 reaction usually we run, and when we run on the gel, so you can see that every one nucleotide change can be separated from another one. So like if size is 100K, 100 base pair, so one will come first like this, second like that. So we need to count band between the that. If A, T, G, C, after A, G, if second band is coming just beneath, so AT, and if another one is came before that one, then AG you can say that. So one single nucleotide differences can make the in the movability of the DNA into the gel. That gel is very thin. Okay. So initially people sometime do for the mutation. Sequencing was not NIH and mistake of the. So that's why machine error. Three or four reactions. Okay. So this round red is called the methylated CpG island, and just only without that red circle, only lines, that is called the unmethylated CpG. So normal cells and cancer cells. So you can see the differences. Tumor suppressor gene with the promoter CpG island, open chromatin conformation. Okay, if that is a hypermethylated, then transcription stopped. So if that particular gene is playing role, some DNA repair mechanism or something, in that case they can become a cancer cells. If epigenetic modification happens in that case. In cancer cells, if something is like in normal condition, something is stopped, but if unmethylated, there is a overexpression of particular genes. So in both cases, methylation of the DNA can cause the cancer. Some so it always dependent on the condition and which gene you are regulating. Okay. So entry, like here already mentioned, entry into cell cycle, avoiding of the apoptosis, defects in DNA repair, angiogenesis, loss of cell adhesion. They can lead to the tumorigenesis. In another scenario, loss of imprinting and the overgrowth, appropriate cell type expression, genome